Right, welcome back guys. This is the second part of the fourth lecture of the experimental design and analysis component of the core skills modules. So before the break I was talking about this idea of statistical probability and it lying at the heart of all statistical tests, and we're going to look at this idea in the context now of a typical experiment or set of observations where we're looking for differences in some kind of interesting biological system, which is what scientists across the life sciences do every day. So I've talked about differences, but really there are sort of broadly speaking two patterns, types of pattern that we are interested in as scientists or as life scientists. We're interested in differences and trends, and these two types of pattern are the sort of bread and butter of hypothesis testing in the life sciences. And just before I sort of start to focus on differences, I just want to identify the fact that there are, broadly speaking, two different ways of visualizing these patterns that we might be interested in, differences and trends. So we would typically visualize data in the form of graphs in order to to spot these patterns perhaps before we do any analysis. So when we're looking for differences, what we would typically do is plot some measure of central tendency for each of the groups that we're interested in comparing. So here I've got two groups A and B, and I've calculated the mean and the standard error and plotted it on a graph, and that gives us an idea of both how the means differ. In this case, Group A has a higher mean than Group B, and also the degree of confidence that we have in each of our two means, which are ultimately estimates of underlying populations, and those are reflected in the error bars as we discussed in previous lectures. So this kind of graphical display of measures of central tendency with measures of confidence, or in some cases we might want to plot measures of spread or variability, is really crucial in the sort of early stages of making inferences from data. We've collected data, you want to look at them to see what sorts of patterns are emerging, probably before you then run tests to establish whether the patterns that you're seeing are likely to have arisen by chance, with the null hypothesis being true, or if in fact we've got enough evidence to reject the null hypothesis. Oh, for these kinds of plots, increasingly it's becoming popular to plot more information than simply the measure of central tendency, the mean in this case, and a measure of confidence, the standard error. It's modern graphics packages, stats packages, including R, for example, the one that I teach to the biology students, and Prism, which the neuroscientists use, and the others that are available, will now plot all kinds of different sort of difference plots. And in many cases you can plot the actual data at the same time as the mean and the standard error, which is what I've done here. The colour scheme's not particularly beautiful, but what you can see here is three different doses of a drug, and we've got the mean as a red blob in the middle. We've got error bars representing our confidence in the mean, but underlying that we've also got the actual raw data, so each dot here represents an individual in the data set, so that we can see simultaneously the measure of central tendency, how confident we are about it, and the variability in the data set, not expressed as a standard deviation or a variance, but expressed as the data themselves, because humans are quite good at seeing patterns in these kinds of plots. With a computer we can provide all of this information instantly. Essentially the whole distribution is there for us to view. So we can, for example, look at outliers, or we can explore the degree of skew in the data set. Do you have a long tail of positive values and all the data squished up down here? So those plots are really useful, and I would encourage you during the course of your degree to think about the way that you plot data and use stats packages appropriately to wring the most out of your data. The other kind of pattern that we're looking for in the sciences is a trend. So that is a relationship between pairs of continuous variables, and those are typically visualized in scatter plots. So here we've got a scatter plot showing a possible relationship between two measurements. Looks like maybe there is a positive relationship here. We wouldn't normally join the dots with a line, and we wouldn't normally put a line through the data unless we'd done some appropriate statistical analysis. When we're at the pattern spotting stage, then we are simply plotting a scatter plot. Okay, so we're going to look more at trends later on in the course, but for the time being we're going to focus on differences, and we're going to imagine that we've collected data like those in the graph on the previous slide, and we want to know now, should we reject the null hypothesis? What is the probability of seeing that difference in the means that we can see if the null hypothesis is true? Okay, so the task here is to compare two or more samples and ask whether they are different. So I find it easiest to visualize these ideas on a frequency histogram. So these two lines, the blue and the green line, represent two possible samples or groups or treatments in an experiment, and we're interested in the degree to which they overlap, essentially, the degree to which these two samples are different. Are they sufficiently different that difference is unlikely to have arisen by chance if they are both from the same underlying population? So there can be two samples in a different study, or there can be many more, and we're going to come on to look later on in the course at more complicated experimental designs where we have more than two samples. But for the time being, in this lecture we're going to focus on the simplest possible situation where we just have two samples, and we want to know, are they significantly different? Do they come from the same underlying population, or are in fact we looking at two different underlying populations? So if you remember, the sample mean is typically denoted x bar, and in this case now we have two sample means, x bar 1 and x bar 2, and we want to know whether those two are different, and we're going to use a statistical test to make a conclusion from the data objectively rather than the alternative, which would be a kind of subjective, have a look at a graph and decide whether or not we think the difference is significant. So this is what stats are doing for us. They're producing objectivity, a systematic standardized way of computing the probability that what we're looking at could have arisen by chance. And we'll do this in the context of a biological example because it always helps. So let's have a look at, this is another neuroscientific example really. We're looking at a drug called calcineurin. We're looking at drugs which inhibit calcineurin, which is a signaling molecule in the body. And we're interested in whether calcineurin inhibitors might have interesting side effects, because actually ordinarily calcineurin inhibitors are used for suppressing the immune system after transplants. So they're a really important drug already in the health service. But the data I'm looking at today come from a study which was exploring a possible side effect of these drugs, which actually might be quite useful. And the side effect is that there seems to be an effect of these calcineurin inhibitors on male fertility. So this is where all this business about sperm swimming speed is building up to. So obviously, if you're not trying to inhibit fertility, that could be a problem. But you'll also be aware that there's been a long search over the last God knows how many decades for good, effective, efficient and safe male contraceptives, which could be taken in oral drug form rather than the alternatives that are available at the moment. So something equivalent to the pill that women are able to take. And calcineurin was potentially a candidate, calcineurin inhibitors were potentially candidates here because they appear to be interfering with male fertility. So in this experiment that we're going to look at, whether calcineurin inhibitors do indeed cause sperm to swim less well and hence perhaps make lower fertility and mean that they could operate as a contraceptive. So we take mice because we need a mammal system to test this idea in that's close to humans taxonomically, so we have some idea of whether it's likely to work in humans. And we treat the mice with calcineurin inhibitors for 2 weeks. And then we measure the sperm swimming speed immediately after the 2 weeks of treatment, and then we wait for 5 days and test them again. And the reason for testing them again in this particular study was because if you're going to look for a drug which could be used as a male contraceptive, you want its effects to be reversible. You don't want to sterilize men for life. That would be pretty disastrous if what you're trying to do is develop a contraceptive. So we're looking for a reduction in sperm swimming speed that is reversible. Hence, we're testing the mice immediately after the treatment and then sometime later. And obviously we need a control group of mice that are treated exactly the same but without the calcineurin inhibitor. And crucially, because we're going to follow the rules of good experimental design, we're going to assign male mice, because we're looking at male fertility, to these treatments, the control treatment and the calcineurin inhibitor treatment, at random. What that means is that the mice in the calcineurin inhibitor treatment group will share nothing in common apart from the treatment. So it's not that they're the first mice that we managed to catch in the cage or the first batch of mice that arrived in the lab. They are truly independent data points because they only share one thing in common, and that's the thing we're interested in, which is the treatment with the drug or the control treatment. So the null hypothesis here is that there's going to be no difference in the sperm swimming speed between the treated mice and the control mice. Pretty straightforward. We're going to collect some data and we're going to challenge that null hypothesis with the objective, if you like, of finding enough evidence to be able to reject the null hypothesis, which might lead us to conclude that we have a potential contraceptive. So the results, let's have a look at them on a graph. Like I say, it's often the first step here is once you've collected some data is to have a look at them visually to look for patterns. And what we've got here is the swimming speed on the y-axis and the data for the 2 time points immediately at the end of the study and then 5 days later. And we've got the 2 groups of mice. In grey, we've got the control mice who were not treated with calcineurin inhibitors, and in black, we've got the mice that were treated. And so what you can see from the graph is that after 1 day, or at the end of the calcineurin inhibitor treatment, it looks like the average swimming speed of sperm from the control mice is perhaps twice what it is in the treated mice, suggesting that calcineurin inhibitor treatment really does reduce the sperm swimming speed, which could then reduce fertility. But remember, we had that question about whether this is reversible. Well, it looks like 5 days later, that difference has more or less disappeared. So that's quite promising. But it would be inappropriate simply to take this graph and write your work up and submit it to a journal and then go ahead and start trying to develop this as a contraceptive drug for use in medical situations, because the inference we've made so far is based on eyeballing a graph, and that's not enough to make a truly objective judgment about whether what we're seeing is representative of a real underlying effect of calcineurin inhibitors, or if the differences that we've observed either at day 1 or at day 5 could have arisen just by chance, given that every mouse will be different and we've got 2 small samples of mice, those that were treated and those that were not. Maybe we just happened to pick for our control treatment mice that had better swimming sperm just by chance, not because of the treatment. That's the question we're having to address with the statistical test. So we want to know whether we should reject our null hypothesis, both at day 1 and day 5. So we're actually going to run 2 tests for the 2 different time points in the experiment. So I'm going to labour this point a bit. We're looking for this p-value, which is going to tell us about statistical significance, and there are lots of ways of expressing what it is we're trying to achieve. We're asking whether the difference between the 2 samples is statistically significant. We're asking what are the chances of observing that difference, the difference that we have observed, if the null hypothesis is really true. Hence, we're calculating a p-value. The smaller the p-value, the less likely it is that the difference we've seen could have arisen by chance in a world in which calcineurin inhibitors have no effect on sperm swimming speed. So typically what we do in science is adopt a sort of threshold below which p has to fall for us to be able to conclude that the difference we're looking at really is significant. In other words, biologically meaningful or representative of a real biological effect. And that threshold has a name. It's called alpha. And you'll see later on why we give it a name in future lectures, not today. So alpha is typically set to be 0.05 or 5%, or maybe 0.01 or 1%, but other values are used. And what we're saying here is that if the chances of the data occurring under the null hypothesis are less than 5%, or maybe less than 1%, we think they're so unlikely that we can effectively ignore that possibility. That must mean the null hypothesis is wrong. Instead, the data must have arisen because the alternative hypothesis is better. And what you'll see in scientific publications, textbooks and papers is these thresholds appearing regularly. And sometimes the p-value is quoted exactly, and sometimes it's quoted relative to a threshold. So for example, you'll see p is less than 0.05. Sometimes this is simply abbreviated to a single asterisk, meaning it's significant. Sometimes we sort of give it a double asterisk if it falls below the 0.01 threshold, and a triple asterisk if the probability is less than 1 in 1,000. And you'll sometimes see NS written to mean not significant if the p-value is greater than or equal to your alpha value, your threshold, which is typically 0.05. So just that hopefully explains that kind of notation, which you're going to see time and again in scientific analysis. So in our case, if p is less than 0.05, there's a less than 5% chance of observing the difference between the two groups of mice that we've observed if the null hypothesis is correct, because the null hypothesis says the drug has no effect. Saying that another way, if we collected the same samples in the same way 100 times, then fewer than 5 times will we see a difference as big as the difference we've seen if p is less than 0.05. Okay, so we're going to look at actually running the test in the next part of the lecture, but now's a good time to break, because what I've just said is this absolutely central concept in all of the lectures really that I'm delivering on this course. So do try to reflect on it and make sure that you're confident in understanding what I've said.